Hello everyone, welcome back to the channel. So today I've got a special treat for you. In case you're unaware, Donnie Badinsky at Standing for Truth and Christopher Rupe, who is a research associate with John Sanford at the Feed My Sheep Foundation, that's actually what it's called, they put out an article a couple of weeks ago now on genetic entropy. And the article was meant to respond to a specific critic, which was Grayson, who never fails to get under Donnie's skin, who made a comment about whether or not genetic load could solve the, quote, mutation load paradox. And I read the article. It was put out in a community post. I left a comment as sort of like a cursory response. I told them in the comment I'd be happy to discuss this issue further with both Donnie and Chris. But then I just kind of laid out what I thought was some issues with the way they presented the history of genetic load. Chris responded and basically accused me of not reading the article very carefully. And then Donnie later responded by putting out a video in which he basically said the same thing. And again, like I said in my original comment, I didn't go all the way through their entire argument in that one comment because, one, you can't like really deal with an article that's what 9 or 10 pages long in a YouTube comment, and that I would probably make a video on this. So here's that video. I've had a couple of people send me his response as a sort of a way to try to, I guess, coax me to respond to it. But we had a historical ice storm here in the Midwest that left me without power for a week. So a lot of this was going on when I couldn't even watch the videos in the first place. So this is going to serve as both a response to the comments, the back and forth with Chris, the response to Donnie's video, as well as a review of their article, right? Because that's really what we should do first. And then we can get into sort of the back and forth that we've been having over this. So let's sort of start off the overview of the critique with the different points we're going to hit as we go along. So first, we're going to talk about what genetic load actually is. Genetic load is a broad concept of which many things fall within it. So we need to kind of understand that historical background so that we can understand what me and Chris were arguing about in the first place, right? Then we'll get into genetic entropy, and then we'll talk about who I'm gonna abbreviate Rupe and Badinsky as RB just for expediency's sake. We're gonna talk about what their thesis is in the paper, and we're gonna critique that. They bring up Haldane's cost of selection, which is part of my original critique of the history, the way they present the history. So we'll talk about that. As well as Kimura's motivations, which was another one of my critiques of the way they presented the history, as I think they misrepresent what Kimura's motivations were. So those two things go into my critique of their history section. Then we're going to get into Kondrashov's 1995 paper, The Contamination of the Genome by Very Slightly Deleterious Mutations. This is a paper I've seen them cite a bunch. And it's one that I was eventually going to make a video on, and I was like, you know what? They bring it up in this paper. In fact, they have a subtitle on one of their sections is The Contamination of the Genome by Very Slightly Deleterious Mutation, like basically the title of Kondrashov's paper. So it seems relevant here that we talk about it. Because quite frankly, Kondrashov's paper does not say what they say it says. And so we're going to get into the math and we're going to show why. Then we're going to talk about Mendel's accountant. This is something that Donnie brought up in his video response. He claimed that it was the most sophisticated program out there, that it's been poorly represented by us. So we're going to talk about Mendel's accountant in some detail. And then we're going to end by just reviewing my previous critique. So again, I'm going to review their thesis. We're going to talk about their history, how I think they're misrepresenting the history. But then I do want to go back to my fatal flaws of genetic entropy video to just hit those major points, because those major points were not addressed by the article, Donnie's response, or Chris. They were things that I mean, obviously, I don't know if any of them watched the video in the first place, so I'm going to use this as a chance to just lay out those points so that they're very clear for them in case they watch this video. Okay, so let's start off with sort of the broad overview of genetic load. So Genetic load, as I said, is a broad term, right? It refers to basically any reduction in fitness in a population that is due to genetic effects, okay? There are multiple types of genetic load, but the ones that we're going to be interested in this video and for this conversation is the mutation load, the drift load, and the substitution load. We're going to introduce the first two here, and then we'll bring up the substitution load when we start talking about Haldane's cost of selection. The first two are the ones that I think are actually relevant to this conversation. The third one I don't think is relevant to this conversation, and I'm going to explain why. Okay, so first, mutation load is defined as the fitness cost to a population due to recurrent mutation. Okay, so it's about mutations that are constantly adding deleterious variation into a population. Selection is removing that variation, but since mutation keeps happening, what ends up occurring is a balance is reached, right? Where mutation is adding in those deleterious variants at the same rate that selection is removing them. And that's what Haldane in 1937 first showed, is he showed that there's an equilibrium balance that will be reached between mutation and selection. And what's really cool is that balance is actually independent of the cost of each deleterious variant on the individual that harbors it. So first off, to understand his model, we need to recognize, as I just said, that mutational load is independent of selection, and this is assuming that the population size is sufficiently large. So in his models, he uses an infinite population. The thing here is like to get rid of the effect of drift, right? So in populations that are very, very big, drift has minimal effect on the population. Right. So basically, when we're thinking about mutational load, we're not thinking about the stochastic effects of drift. We're thinking about the deterministic effects of mutation constantly adding back in that deleterious variation. So he derived that at an equilibrium frequency, that is X, is that equilibrium frequency, it's equal to the mutation rate divided by 1 minus the fitness of the individual that's carrying that deleterious variant. Okay. So what he found really, really interestingly is, as I said, the equilibrium frequency that is reached, and that equilibrium frequency is also, you can think about it as the cost to the population for carrying that variant, is virtually independent of the cost to each individual, right? So I've plotted this on the bottom right here. So on the x-axis is the fitness of the population, or the fitness of the individual that's carrying that deleterious variant, right? So as that fitness gets higher and higher and higher, then you start to deviate more from the dashed line. And what that dashed line represents is just the mutation rate, okay? So at equilibrium, we expect that the rate at which those variants should be in the population is exactly equal to the mutation rate. Now you can see it does diverge above it as the fitness of the individual carrying that deleterious mutation gets higher and higher and higher. But if you look at the y-axis, it's very, very small, right? We're still in the 2e to the negative 8 territory where the mutation rate's 1e to the negative 8, right? So it's a very, very minimal increase. But what this shows us is something really, really cool, right? Is that it doesn't matter if the mutation is lethal, right? If individuals that harbor it literally just die, or if the mutation is virtually harmless, right? If it's very, very, very mildly deleterious. Either of those don't matter, right? Then the frequency will still be around the rate of mutation. That's what Haldane showed. So then, if that's true, right, if the rate at which you should find that deleterious mutation, given this mutation selection balance, is at the mutation rate, therefore the cost to the population, the fitness cost, is exactly equal to the mutation rate, right? So to give some numbers to this, if the mutation rate is 1e to the negative 7, then the fitness of the population is 1 minus 1e to the negative 7, which is 0.999, right? It's a very, very minor decrease in fitness. And as I said, it doesn't matter how deleterious the variant is. Now, again, here we're talking specifically about very large populations such that the randomness of drift is virtually removed, right? You virtually don't have any randomness of drift. The only things at play here are mutation and selection, right? And they reach this balance, this equilibrium frequency. So that's the historical context for the mutation load. And like I said, it goes all the way back to Haldane in 1937. Now let's talk about the mutation load paradox. So this is the observation that the deleterious mutation rate is so high, we shouldn't exist based on our theoretical understanding of the mutation load that we just derived. Let's talk about this in some detail. So first, Moto Kimura and Takeo Murayama in 1966 showed that the load could be approximated as 1 minus e raised to the negative u, where u is the average deleterious mutation rate per genome per generation, right? So let's think about what this means, and let's see if we can get some estimates for this in humans to see what the estimated genetic load or mutation load is in humans. So this study did something pretty cool to try to get an idea about how many mutations are deleterious. So what they did is they measured the mutation rate for transposons. What is the mutation rate for transposons in the human genome? And then they compared that rate to the rates of mutation for non-transposons, right? And they found that non-transposable elements in the genome evolve at about 94.3% the rate of transposons. So what that means then is 5.7% of the mutations that are occurring in non-transposable regions are being removed. They're deleterious enough that they're getting culled by selection. So if that's the case, right, then that's what's relevant to the mutation load, is how many of those new mutations every generation are actually deleterious enough for selection to act on them. Well, that's 5.7%, okay? So now let's look at some numbers given an idea about how much of the genome is functional, and how many mutations you get per generation. Okay, so let's say 10% of the genome is functional, and you have 100 new mutations per generation. 0.1 is the fraction that's functional, so it's 100 times 0.1 times 0.057. The 0.057 comes from the 5.7% that were estimated to be deleterious. Then we just plug in our numbers, and our load ends up being 0.43. What does that mean? How do we understand that? Well, for one, that means that 43% of the population on average won't have offspring. Okay. And while that seems really high, let's think about what that means in terms of like reproductive cost. So the cost of reproduction then is 1 over 1 minus 0.43, which equals 1.7. That's how many offspring females need to have. Now, since we're a dioecious species, females need to have enough to replace not just themselves, but to also replace the males. So they need to have actually twice that number. And so 1.7 times 2 is 3.4. So if, on average, females have 3.4 offspring, then there's no paradox, right? Then the mutation load that exists in humans, given those conditions, is tolerable. And 3 offspring is well within the historical range of the number of offspring females can have over their lifespan, right? So again, 10% of the genome is functional, with 5.7% of all new mutations being deleterious. There is no mutation load, or there is no mutation load paradox, I should say. What if we increase that number to 25%? So 25% of the genome is functional. Now what we get is a load of 0.76. 0.76 means 76% of the population does not reproduce. So now how many offspring would females need to have to make up for that cost? Well, they would need to have, on average, 8 offspring to make up for that cost. 8 is way higher than what the average number of offspring is in humans. That's a really big number, and most women don't have that many children, right? So what that means is that introduces a paradox. And we'll talk about what we mean when we say it introduces a paradox when we go through some more of the numbers. So what if we increase it to 50%? So at 50%, the load is now 0.94, which means 94% of individuals aren't reproducing, which means that for females to keep up with the population size, they would need to have 33 offspring. Humans don't have that many, right? If we go all the way to 100, that's a load of 0.99. That's 99% of individuals don't have offspring. And so females would need to have 200 offspring to make up for the cost. Okay. So, and it should be noted that all of these are relatively conservative estimates. So the 5.7% are deleterious. That's a conservative estimate based on, this paper sort of napkin calculations. Other studies have found that the deleterious mutation rate can be high as 10 deleterious mutations per individual per generation. Again, if it's 10, right, I've plotted this down here on the bottom from 0 to 10 on the x-axis and then the proportion of deaths on the y. You can see basically for values above 4, nobody is surviving to reproduce, right? At values of 10, 99.99999, like virtually nobody is having offspring. So what does this mean? What does this mean broadly, and what does this mean in terms of like the mutation load paradox? So importantly, what the mutation load paradox represents is not mutation load. That's not what this is saying. This is not saying that this is actually what humans are suffering. It's saying that we can't actually be suffering this or we wouldn't exist, right? If the load, for example, were 0.76, 76% of females every generation would have no offspring. That's not true. We know that that's not actually what happens, right? And females are certainly not having 8 offspring per generation. We know that's not happening. So that's why it's a paradox. It's a paradox because the theory seems to suggest that it's something that should be existing, but reality doesn't, right? And so when we start talking about resolutions to the mutation load paradox, the way that RB kind of pitch it is as if these are rescue devices, like we're trying to save the human population from extinction due to the mutation load paradox. That's not what we're trying to do, right? Because if the mutation load paradox were real, if it really were a mutation load, we would be dead already. The population wouldn't even exist. We would have been dead generations ago, right? Like we wouldn't even be around. So very, very importantly, mutation load paradox does not equal mutation load. Just because there exists this concept we call the mutation load paradox does not mean that humans are actually suffering from it. Again, the mutation load paradox is not about we are slowly winding to extinction. That's not what it says at all. It's that every single generation, females would need to have 8 to 200 offspring, depending on what the deleterious mutation rate you think it is, just to persist, just to maintain a constant population size. That obviously doesn't exist. And so when we start talking about resolutions to the paradox, what we're really talking about is trying to find where our theory fails. What are the holes in our current understanding of the theory or of reality, of biology? What are we missing in real systems that is not being accounted for in our models of mutation load? That's what we mean when we say resolving the paradox. And that's why you hear a bunch of different ideas thrown out there to try to resolve this issue. And it's about taking theory that is itself just theory. And theory is only as true as it can explain reality. This is very important. You can have the most elegant math ever. It can be beautiful, elegant, but if it doesn't match reality, it's useless. Right? It has no purpose in biology if it doesn't match reality. And right now, the mutation load paradox doesn't match reality. So we need to find a way to solve it. And basically, we're solving the theory. We're not trying to solve reality, right? They've got it backwards here. So bear this in mind as we go forward, and then we'll kind of see the way in which they're coaxing the mutation load paradox as if it's a real paradox, as if it's not a paradox, but a real thing. Okay, so now let's talk about drift load. So drift load is the fitness cost to a population due to the accumulation of weakly deleterious variants. Now, as opposed to the deterministic load that we were just talking about, this load is stochastic, and it's due to the randomness introduced because of drift. And I've pictured here Tomoka Oda because she is actually the founder of the theory of nearly neutral evolution. She is the founder of this. She was Kimura's grad student. She does not get enough credit for this. Nearly neutral theory is her idea, not Kimura's. Kimura was mostly focused on strictly neutral allele. So I'm giving a shout out to Tomoka Oda because she doesn't get enough credit. Okay, so one thing to remember is that when we're talking about mutations being added into the population, selection removing them, for selection to be able to remove those mutations, it has to overcome what's called the drift barrier, okay? And that means that it has to be stronger than the rate of genetic drift. All right. If it's not, if selection is weaker than the rate of genetic drift, then the frequency of any deleterious variant will just fluctuate randomly. Right, because selection is not strong enough to actually remove it from the population. You can think about it as drift weakens selection, right? So the smaller and smaller and smaller your population is, the more those frequencies bounce around by drift, and so selection just can't keep up with the stochasticity of drift. All right, that's the drift barrier that has to be overcome. And what this means is that there's a parameter space that exists where there exists a drift barrier. Okay, I plotted this down here on the bottom where you have the probability of fixation on the y and then the strength of selection on the x. The dashed line in the middle is the strict neutrality. And you can see that there's a clear difference based on population size as to where that drift barrier is. So if your population size is very small, in the green that's n equals 500, you can see the probability of fixation is still pretty high relative to like the rate of the initial frequency compared to when n is equal to 10,000, right? Or when n is equal to 10,000, as soon as you get slightly deleterious, the probability goes basically to zero. Right. So as the population gets larger and larger and larger, the number of deleterious variants that don't overcome that drift barrier gets less and less and less. Right. And this is because, again, selection is getting more efficient as the population is larger. And then collectively, they can reduce the population's fitness. And so this is the load part, right? And so here load is equal to 1 minus s, where s is the selection coefficient. Remember, it has to be some value less than 1 over 4 n sub e, where n of e is the effective population size. So for drift load to be true, s has to be less than that number, and then raised to the g, where g is the number of loci that have been substituted. So, if you're reducing it by some percentage, right, if s is like 1e to the negative 5 or something, and you have 20 of those loci fixed, then g is equal to 20. All right, that's how you would calculate load in this context. And as I said, drift load exists in a narrow parameter space. And drift load becomes negligible whenever these two things are satisfied. That is that selection is less than 1 over the genome size. And by genome size here, we mean the functional fraction of the genome. So the part of the genome that can actually have a deleterious mutation of any kind, right? That's that fraction we're talking about there. And this is really important to understand. So selection cannot be infinitely weak. Right? It can only get so weak before it no longer matters, before you could substitute every single site across the entire genome with a less fit variant. But if the cost of fitness of each of those variants is infinitesimally small, then you're not reducing the fitness at all. I'm actually going to show that math a little bit later on, but just bear that in mind that when they try to stress, RB try to stress as very slightly deleterious, infinitesimally slightly deleterious. No. It still has to be greater than 1 over the genome size, or it doesn't matter. You can substitute every single site to a less fit variant, won't matter. And as you would just intuit from this, these are most relevant to organisms of conservation concern. Like cheetahs, when you're talking about like rhinos, like these organisms that are well known to have very, very small populations that we're concerned are going to go extinct. They tend to harbor a much greater drift load because selection is very weak on small populations, right? That's the idea. And I mentioned this in my back and forth with Chris that drift load is most relevant to conservation concern. It's not relevant to organisms that have very big population sizes. Right, so you can't invoke genetic entropy as this like thing that affects everything, right? Like everything is winding to extinction because of the accumulation of very slightly deleterious mutations, when probably the majority of living organisms don't actually exist below the drift barrier, right? Especially not like invertebrates that have enormous population sizes. So just bear that in mind as we go forward. Okay, so we now come to RB's article, now that we have some of that historical background out of the way. First, we need to understand what genetic entropy is and why it is fundamentally different than the two things we just talked about. It's fundamentally different from genetic load. Genetic entropy claims that all genomes are winding down to extinction from an originally perfect state due to mutation accumulation, okay? And that's what they write here. This is a screenshot from their article. They said that it is based on the observation that the mutation rate is high, that most mutations are harmful. Then they say yet not harmful enough to be removed from the population by natural selection. And specifically, RB write, quote, the process of mutation accumulation began when the first man, Adam, sinned, resulting in the entrance of death and a fallen creation. And this is very interesting because, again, it insinuates that there was some original genome, just Adam's genome, that has ever since been degrading. All of his pedigree ancestors, their genomes have been degrading ever since. And this is a specific claim, and we can test this claim. So what this means is that across the genome of every single human being, then we should all have the same coalescent ancestor that existed at the same point in time. Because Adam is that original ancestor, so as you go across your genome, remember, your genome is a mosaic of ancestries. Recombination is breaking up associations between nearby haplotypes, which leads to each haplotype having its own independent pedigree, right? And so all across your genome exists hundreds of ancestors. Not one ancestor, and definitely not one ancestor living at one point in the past. There are hundreds of ancestors that lived at all different times in the past. And I'm showing this on this figure down here from Rasmussen in 2014. This is just one small region. This is not even like the full genome. This is just a portion of one chromosome, chromosome 6. And notice first, they have annotated where the gene regions are. So these are the actual protein coding genes in the dark blue over the top. And then down below on the y-axis is the time to the most recent common ancestor, and then the x-axis is just genomic position. And so notice, as you go across your genome, the time to the most recent common ancestor, when each of those regions find a common ancestor, is different. Some is really deep, some is really shallow. It is different everywhere you go, which means all across your genome, there are different ancestors. There was no one first man that existed. Another cool thing that I really want to point out from this study is notice how in the regions where there's very gene dense, so a lot of those blue lines, so these little areas here, this is a very gene dense region. This is, dense gene. Notice how the time to your most recent common ancestor is very shallow in those regions. What this represents is selection. So anytime you have any individual that might have been in your pedigree that has a mutation in that region, selection gets rid of it. So that individual ends up not being part of your ancestry, right? They don't pass that mutation on. And so what this means is that selection is constantly sort of culling your pedigree. And so for regions that around functional areas of the genome, they have very shallow ancestries because of selection culling those regions. So just wanted to let you know that that's what you're seeing. And that's why the areas with the largest peaks are those that are falling between those functional regions because they're not under any selective constraint. Mutations can happen all the way back to some distant ancestor, and selection is not getting rid of those ancestors from your pedigree. Very simple claim. There was one original genome, one original person that was the ancestor of everyone. This very clearly shows there was no one ancestor. There were many, many ancestors, and they all lived at different times. So every single individual that's an ancestor in this genome was a member of a population, and that population had unique variation that has been differentially lost and fixed in different parts of the genome. Okay, next we move on to their section, Natural Selection to the Rescue. But what exactly needs saving? Okay, so they write that critics of genetic entropy have long suggested that natural selection can come to the rescue by removing most of these harmful mutations from the population and amplify any rare beneficial mutants. These critics insist that selection is efficient enough to overcome perpetual fitness decline, but is this feasible? So expectation, given what I just told you, given what I just taught you about sort of the history of mutation load and drift load, what you expect them to talk about is, can selection prevent mutation accumulation, right? Can it overcome the drift load, right? That seems to be what they mean by natural selection to the rescue. What we get, though, is this argument that there's a cost of fitness when an adaptive allele spreads through a population, which means that they are taking a detour into a different idea called the substitutional load. So we're going to follow them through this rabbit hole and talk about the substitution load. So substitution load is the fitness cost to a population as a new allele is increasing in frequency. Okay, this was first derived by Haldane in 1957, 20 years after he derived mutation load, mind you, and in a paper called The Cost of Natural Selection. The best way to sort of think about this is actually via a little analogy, and it's the same one that he used. So imagine, for example, you're a cattle rancher, and there's some new variant in your herd, and you want that variant to spread and for everybody in the population to have it, right? Well, when that variant is very rare, you can't just like cull your entire herd, right? Like generally what you would do as a breeder is cull the individuals in the population that don't have the variation you want and only breed those that do have the variation you want. Well, when that variation is very rare, you can't do that because it needs to spread first, right? So this kind of like sets a limitation on how quickly this particular allele can spread, and it demonstrates that there's a cost for this allele spreading, right? So if you think about it, how much of their herd can you possibly cull to allow that allele to increase in frequency, and how many offspring must that rare mutant have to compensate for your culling, right? So that's effectively like the idea of Haldane's cost. I'm showing the math down here. You can see that across the x-axis is the frequency of that variant that's spreading through the population. It starts off at a frequency very, very small, and then eventually goes to fixation, right? And then on the y-axis is the selective deaths, the number of deaths that must happen for that allele to increase in frequency, right? And so you can see when the frequency is very, very low, as I said, the number of deaths for it to start increasing is very high. That individual needs to have a lot of offspring to compensate for the reproductive loss of fitness of most of the population. And then eventually the cost goes down, right? As that variant spreads and spreads and spreads, it doesn't need to have as many offspring because most of the population is represented by it. Okay, so basically what this does is this places a limit on how fast an allele can spread through a population, right? You can't just like take that initial beneficial variant that you want in your herd and just kill everybody else, right? So you can't, you have to slowly let it arise. That's the idea. That's substitution load. It's about an adaptive, a beneficial variant can't spread just infinitely fast in a population. That's it. There's nothing like crazy about the idea. It was not seen as some like unbelievable fault of evolution or anything like that. That's not the way that it was seen at the time. But let's see the way they pitch it. So RB introduced Haldane's cost, and they're doing this to create a sort of false problem, what they call evolution slowness. And in doing so, they muddy both selection and mutational load. So they write, quote, "In light of the limited reproductive capacity and the high reproductive cost of selection in mammalian populations, how can there be sufficient number of progeny available for selective elimination every generation, as would be needed to counteract the high rates of deleterious mutation accumulation?" So notice what they're doing here. High reproductive cost of selection is about adaptive evolution. That's the substitutional load, right? High rates of deleterious mutation accumulation is about the mutational load, which is independent of selection. Remember, here's the graph again, down here at the bottom. Selection and mutation will reach a balance that is exactly equal to the mutation rate, which means that the selection term can just be discarded. So they're using these two things, they're conflating these two processes, and they're muddying the waters. They're trying to make it seem like evolution's too slow because of Haldane's cost, so it can't possibly like work, right? When that's not even what mutational load is about. It's not about the speed of evolution, it's about the strength of purifying selection. And they're just conflating these two things, and this is a bait and switch. And they've baited us with this idea of there being a slowness to evolution because like selection can't just act super quickly, but then they now switch to that being the problem with mutation accumulation. That's not the problem with mutation accumulation, right? That's about an allele spreading. Like purifying selection is not about the cost to the allele spreading. It's about just the cost to harboring it in the population. And at mutation selection balance, it should be harbored at exactly the frequency of the rate. Right, so that's why I said in my comments, my back and forth with Chris Rupe, that this was a bait and switch. Right, you're baiting us with Haldane's cost, and then you're switching to mutational load, and you're trying to equate those two ideas as if they're the same thing, but they are not the same thing at all. And then they go on to quote mine Haldane to make this argument about the slowness of evolution. They quote, first, Haldane calculated the time required for a single substitution is equivalent to roughly 300 generations. That's not what he estimated at all. That 300 generations number was just a guess. It was a guess based on what he thought was a tolerable load for mammals. Like that's it. Right. He thought that for the load for at 300 generations is like 0.1. So the beneficial mutation would, anybody that was harboring it would need to have around 10% more offspring to compensate. Right. And that the spread, the rate of that spread was about 300 generations. So that's a guess that he just threw out there into the ether, was a kind of guess that he made. He didn't have any data. He had no idea how diverged organisms actually were from each other, right? So he couldn't have been calculating the time required for a single substitution. That's not what that number represents. They further go on to say he concluded that the process of genetic fixation occurs too slowly via positive selection to accomplish any meaningful evolutionary change. He does not say that at all. That is not what he says. They quote him here. Haldane emphasized in his paper, even the geologic timescale is too short for such processes to go on in respect for thousands of loci. Let's see the context in which Haldane's actually writing that. So below this is the paragraph from Haldane's paper, and let's read it in its entirety. So Zuner in 1945, after a full discussion of the Pleistocene fossil record, concluded that in mammals, about 500,000 years were required for the evolution of a new species, though in some voles genera the rate was somewhat greater. Some insects seem to have evolved at about the same rate, while other insects and all mollusks have evolved more slowly. Again, that they're using the fossil, they're basing this off of just morphological change in the fossil record. He estimated the total duration of the Pleistocene at 600,000 years, but some later authors would half this figure. On the other hand, environmental changes during the Pleistocene were unusually rapid, and evolution, therefore, probably also unusually rapid. Then notice what he writes here. The agreement with the theory here developed is satisfactory. Remember, like, this is just gradualism. Right? They were just looking into the fossil record and saying that, oh, it takes a few million years for these changes to happen, for like visible morphological change to happen, doesn't happen immediately. It takes some time. And so in his mind, the theory he just developed is exactly matching that. He's not saying that evolution is too slow. He's thinking that the theory he just put forward is actually well matched by the fossil record. Remember, he has no genetic data. It's just based on a guess of the amount of change given the fossil record and a guess about how many loci that might be contributing to that change. They did not know at the time. Okay, so where does that quote actually come from? So he writes, some writers, such as Fisher, appear to assume that the number of loci at which modifiers, for example, genes affecting the dominance of other genes, may be selected is indefinitely large. Right? So Fisher's idea about natural selection was almost mystical. He believed that natural selection could work independently everywhere, that it was the most efficient thing, and that drift could always be ignored. Fisher was like a proselytizer for natural selection. Haldane is critiquing that idea. Right? He says that the number of loci is finite, and that the cost to the substitutional load would be too high. Like, you couldn't just have selection acting indefinitely on every possible loci like Fisher thought that it could. That's what he's referring to here. He's referring to the fact that thousands of loci could not have been selected over that time because it would be too big of a substitutional load. Now remember, again, he didn't know how many loci are actually contributing to the divergence between organisms. We know now that it's actually not that much, that the functional fraction of divergences between like our protein coding genes are not that diverged from one another. A lot of the divergences between organisms are not in functional regions and are not positively selected. Which leads us to the next point. RB claimed that Kimura developed neutral theory to solve the slowness of evolution. They write, brilliant mathematician and renowned evolutionary population geneticist Kimura recognized the severity of Haldane's cost of selection. It was because of the slowness of evolutionary rates of substitution that Kimura championed his neutral theory of molecular evolution. This is false. This is just false. That's not at all why Kimura derived neutral theory. He did not think Haldane's cost of selection was some serious thing that challenged evolution. This is an incredibly ahistorical take. What Kimura was actually recognizing is that, and to remember, Kimura's work came after Lewontin and Hubby were first estimating natural levels of variation, right? Remember, Haldane didn't have any data, but now in the 1960s and 70s, we start to get genetic data. We start to get polymorphism data. And so what Kimura did is he looked at the polymorphism data and he found like, wow, there's actually more divergence here than what we thought. It looks like the rate of divergence between organisms is actually too fast, not too slow, right? It's not that they're diverging too slowly. They're diverging a lot quicker than what Haldane thought they could. So what Kimura points out is that most of the divergence must not have been adaptive, right? Because if it was all adaptive, if that much divergence was adaptive, then the substitutional load would be too high, right? Because that would mean that every single one of those alleles would have to be spreading by positive selection. But if they're not spreading by positive selection, if they're randomly fluctuating by drift, such that 2 times the population size times S is less than 1 for most of those fixed differences, then there's no substitutional load. Kimura was not trying to solve the slowness of evolution. He was trying to figure out why evolution could happen so quickly. What could possibly be letting evolution happen as fast as it was happening? And his solution was that, look, it would appear then that most mutations are actually not beneficial. They're not being positively selected. They're not deleterious. They must just mostly be neutral. So finally, drift is not faster than selection. And they try to build this case that Kimura was doing this because drift makes evolution faster. Drift does not make evolution faster. Drift is much slower than selection. All drift allows is that for things to fix randomly, so it doesn't matter if there's a ton of divergence between organisms. All that's reflecting is how much standing diversity there might have been in a population, right? And then you can just have random fixation. It no longer matters whether or not there was a selective cost to the fixation of each of those alleles. That's the point of Kimura's neutral theory. Arby's thesis then relies on you not seeing the bait and switch. Basically, they're arguing that selection is too slow and drift leads to mutation load, right? They write, interestingly, Kimura's solution creates a catch-22 for evolutionary theory. Kimura's solution to this non-existing problem of the slowness of evolution. Even assuming that the rate of neutral evolution via genetic drift solves selection's slowness problem, which doesn't exist, it creates a far worse problem relating to the genetic fixation of effectively neutral, slightly deleterious mutations, the implications of which are devastating for evolutionary theory. Again, so that I'm perfectly clear, Haldane's dilemma was about the speed of adaptive evolution, not the strength of purifying selection, not whether or not selection can deal with deleterious mutations. It's about the speed of adaptive evolution. That's it. All right. Purifying selection has not been discarded, and evidence for purifying selection is extensive. Nor have we discarded compensatory mutations. Haldane's dilemma has nothing to do with those things. I've plotted down here on the right just a graph where the blue bars represent exonic regions. The x-axis is just the genomic position. Y-axis is levels of diversity. Notice that purifying selection is removing mutations from those regions that are functional, where exons are, right? That purifying selection has not been discarded. So they're trying to say that natural selection just doesn't work. It's too slow. It's too inefficient. We can get rid of it. No, that's not what Haldane's dilemma was about. That's not what Kimura was trying to show. Purifying selection is still very much a thing. Lastly, is that there are no broad category of unselectable mutations. Okay, so again, they say The worst problem, that creates a worse problem relating to the genetic fixation of effectively neutral, unselectable mutations. Remember all the way back for the drift load, right? Unselectable mutations is a relative term. It's relative to the population size. Some populations will basically have no unselectable mutations. Some will have many unselectable mutations. It's dependent on the population size, which is antithetical to what genetic entropy assumes, which is that all organisms, irrespective of their population size, should be winding down to extinction. RB paint drift load in very broad strokes, and they miss exactly how narrow it is. They write, for example, as a result of this ongoing process, this accumulation of deleterious mutations, the human population must be moving toward extinction due to the contamination of the genome by these very slightly deleterious mutations. Recall what nearly neutral mutation means. Right, as I just said, it means that S is less than 1/4 NE. As NE gets larger, mutations that are nearly neutral become less as selection overcomes the drift barrier. And furthermore, a threshold exists below which, if S is so small, it ceases to matter. This is what we talked about in the drift load, that if S is less than 1/G, it doesn't matter. The effect is too small. For example, if S is equal to 1e to the -10 and the genome size is 1e to the 8, then the cost of substituting every single base pair in the genome, every single one of them is being substituted to a less fit variant, the cost is only 1% fitness. So we cannot paint this in very broad strokes. There is a narrow parameter range that must be satisfied for these things to work. Okay, so the next thing that they bring up, or the next section of their paper, is the contamination of the genome by very slightly deleterious mutations. This is just literally the title of Kondrashov's paper. Kondrashov's model is the drift load model, right? It's the one that's concerned with this accumulation. I've actually showed the model and the model's parameter space down here at the bottom. The little triangle that's highlighted, that's the parameter space, that little triangle, right? Those are the values in which this fall within. So this is the range of parameters. If this is satisfied, then you have the drift load. 1/G is less than S, so S is greater than 1/G, and S is less than 1/4 times the population size. If that is satisfied, right? If that parameter space is satisfied, then you can have a drift load paradox emerge. And so Kondrashov derives it in a way that we can be familiar with, as we did before, with the 1 minus e raised to the -u, as 1 minus e raised to the -g f s. G being the genome size, f is the functional fraction of the genome size, and s is the selection coefficient against each one of those variants. So this introduces a big question, right? Are humans within this parameter space? Do we actually fall in the parameter space of 1/G is less than S is less than 1/4 NE? Well, the human population size is around 8 billion individuals. Our genome size is 3.5 billion, indicating that N is greater than G. If N is greater than G, then we are not in the parameter space of Kondrashov's model. We are not in that little triangle, which would indicate that there is no drift load paradox. Now, importantly, we need to understand something about effective population size. What is actually meant by N of E? So if the effective population size of humans is probably some number less than this, then we need to think about what is the relevant population size value, right? And we also need to think about what's the relevant G, right? What's the relevant G value and what's the relevant effective population size? If we take the inbreeding population size of humans, which is based on events long ago, right? It's the long-term inbreeding population size. We're only 10 to 25,000 individuals, very, very small, and we would be within that triangle, right? Then the drift load paradox exists. If, however, the value of the effective population size that's more relevant to selection today is the variance effective size, then we are not in that parameter. Let me explain what the variance effective size is. So again, the long-term inbreeding size is the equilibrium value of diversity based on deep time events, right? That's the long-term inbreeding effective size. The variance effective size, however, is defined not by the amount of standing variation, but how much the frequency of every variant changes per generation. So you can imagine in very, very small populations, the change in frequency per generation is really big, right? You have a large variance in frequency. If the change in frequency per generation is very small, right, then you have a very large population size. Humans, I think what's probably the more relevant value to selection is not the deep time inbreeding population size, it's probably the variance population size, right? Because selection is acting per generation on the variance in frequencies, right? That's where selection acts, not way back in the past. The way back in the past dictates how much standing variation there is for selection to like act upon, but it doesn't determine the rate at which selection acts or the strength at which selection acts. So instead, I think the variance effective size is what matters to humans, and our variance effective size is probably a lot closer to our census population size, which would again put us outside of the range of Kondrashov's model. Again, Kondrashov's model doesn't apply broadly. There is a narrow little triangle where things can fit within, and it's unclear if humans even fall within that parameter space. Okay, so let's summarize RB's thesis and the points thus far before moving on to Donnie's video. So first, they claim that natural selection is too slow to stop the accumulation of deleterious mutations, and that it is blind to nearly neutral ones. Their slowness argument relies on a misuse of Haldanes substitution load. Selection in this context needs to purge variants. It doesn't need to substitute them. Haldanes substitutional load is not relevant to this because, again, we're not substituting variants. We are purging them. Mutation load paradox does not equal the mutation load, or we'd already be dead. If there really was a mutation load, we would be dead. And this is also true in Kondrashov's model, I should say. If Kondrashov's model is true, that there is a drift load paradox, we would also be dead. Okay, so it's not, neither of these models are about us winding to extinction. They are both, if the paradox exists, if that were true, if the paradox was real and not a paradox, we would already be dead. Kondrashov's model is limited to a narrow range of parameters because drift load exists within a narrow range of parameters, and it's unclear if humans even fall within it. And most organisms don't fall within it. So it can't even be used to describe genetic entropy, which is supposed to paint these really broad strokes across all life as degrading, but they have yet to put forward a mathematical model that could actually show that happening, irrespective of selection, irrespective of population size. That is what I have been asking for from the very beginning, is for someone to please, dear God, give me a mathematical model, okay? Don't hand wave away these verbal arguments. I want a mathematical model that shows, irrespective of population size, irrespective of the strength of selection against each deleterious variant, all populations will go extinct. That's what I'm asking for. And as I literally just said, there's no mathematical model of genetic entropy presented. Genetic entropy needs to demonstrate how drift load would still occur if S is greater than 1 over 4NE or when S is less than 1 over G. Right? You need to show that genetic entropy is not just limited to the parameter space of drift load. If it is, if your argument is that it is, but you think humans fall within that, genetic entropy is not a term you even need. Just call it the drift load, and we'll talk about drift load. Like we don't need to coin a new term for a concept that's existed for 30 years. Okay, so now let's move on to Donnie's comments on comments video about my back and forth with Rupe. He says a couple of things that I want to address here. First is that he misrepresents my comment. He states this, and this is a direct quote, Zach, he's the PhD, doesn't want to acknowledge fixation of very slightly deleterious mutations. Now, you know that's not true, Donnie. That is not what I said. And I've got my comment right here. You can't see it on his screen because it's so small, but here's my actual comment. This is what I say. Mutational load is different, I'm referring to like how mutational load is different than substitutional load, and is related to the rate of mutation relative to the effective population size. This has been observed in real systems as the proportion of deleterious variation being harbored. So I'm literally saying that there is deleterious variation in every population. And I will say here that those variants can fix. They can and they do fix. That's not even what we're debating right now. I go on to say, but it's only ever relevant to species of conservation concern. I absolutely acknowledge the fixation of very slightly deleterious mutations. That's just not a true statement at all. Next, he says this about Mendel's accountant.
As myself and others have shown, incorporating even exceedingly rare beneficial mutants with very small selective advantages are enough to overcome the mutational load for moderately sized populations. No, that is not the case. Firstly, Zach, you said you would like to see a mathematical model of genomic degeneration using a distribution of fitness effects. This has been done. This has been used. This is Mendel's accountant. Paper after paper, article after article has been published using Mendel's accountant, the most sophisticated numerical simulation of evolutionary processes that exist.
Okay. So the claim, Mendel's accountant has been hailed as the most sophisticated simulation software. Is that true? Well, let's take a couple of measures to compare. Okay, let's compare a couple of things and see just how true that could possibly be. So these are a couple of the most popular forward time simulators. You don't even need to know the difference between backward and forward in time. Forward in time simulators tend to be a lot more complex. They're a lot more computationally wieldy, and Mendel's accountant is a forward time simulator. So let's compare the level of sophistication as a measure of impact on the field, right? If Mendel's accountant was really as sophisticated and wonderful as they seem to make it out to be, let's see if it lives up to the expectation that these other simulators have done. So the first one I've listed here, this is the one that I found with the most citations. This is SLIM 3, and it's been cited, as you can see here, 498 times. 498 times, and it was published in 2019, right? So that's a heck of a lot of citations for only being out for a few years. Next one on the list is EasyPop. EasyPop was one of the first forward time simulators, and it was put out in 2001, and it's been cited 407 times. So that's still a good number of citations, but notice it's been cited less than SLIM, which is much younger, right? So SLIM is spoiler, SLIM is the most sophisticated one, but like that's why it's been cited as much as it has. SimuPop is another one that was around at around the same time as Mendel's accountant came out. So both EasyPop and SimuPop came out around that same time, as did Splash for that matter. Notice the SimuPop has been cited 349 times, and Splash has been cited 157 times. So all of these have been cited over 100 times, all over 150 times, some of them over 400 times. So let's compare that to Mendel's accountant's citation record. 49 times. 49. It came out in 2007 in Scalable Computing, a computing journal, not even a biology journal, a journal completely just computer programmers read. In 2007, it's been cited 49 times. When you go through those citations, when you actually go through those citations, the vast majority are cited by Sanford in creationist journals. The only two that actually use the program and that were cited in secular journals were by Sanford. All of the rest of the ones that cite this program that are in secular journals don't actually use the program. Most of them are just listing it because they're like reviewing the literature of forward time simulators, or they're introducing a new simulator and they're just citing all the other ones that existed before, right? They're not actually using the program. If the program was the most sophisticated and was so great and so wonderful, why is only Sanford using it? Why does no one else use it if it was so great and so sophisticated? Well, here's one possible reason. So this was a surprise to me, and this was something that Chris Rupe said in our back and forth. He says, because I was having some trouble downloading, like I wanted to use Mendel's accountant, tried to download it, and was like running into like some firewall problems. So I asked him if he had some suggestions on how I could get the program, and he said this: I have been discussing with certain others the possibility of making Mendel's accountant publicly available again. That phase of research has ended some years ago. However, due to growing interest, I think it is a good idea to make it available again. It's not even publicly available. How can you cite this program as the definitive proof of genetic entropy? This is your model of genetic entropy, and no one can test it. It's not publicly available. Every one of these programs are publicly available. You can go back and download EasyPop from 2001. You can still get it and still run it. Every one of these are publicly available. They have GitHubs. You can see their source code, everything publicly available. Why is Mendel's accountant not publicly available? That needs to be done first. If you're going to say Mendel's accountant is the thing we all should be using, and why aren't we all using it, and we're, crappy for not using, you need to have it publicly available. It's as simple as that, right? Because I wanted to use it, I wanted to assess your claim, and I couldn't. I couldn't replicate your research. Right? This is how science works. I need to be able to see if I can replicate it, because it's not being replicated when I run this on any other simulation software. This is what I said to Chris, is that when I run it on Slim, it doesn't give me those results. So what's the problem? Why does Mendel's accountant not give the results? I wanted to run it and try it out. It's not even publicly available. I can't even run it and try it out. So the next thing I want to do is compare it not just by whether it's publicly available or its citation record, but let's compare by its features. Okay, just the features that it has. First, I'm going to show you the features in Slim. Slim has a manual that is 700 pages long. I'm just going to scroll very slowly through the table of contents for you so that you can see all of the different things that you can do in Slim. And then we'll compare that with the features that exist in Mendel's accountant. Here's Mendel's accountant's features. I could fit it all on a single slide. There are 8 features that it saw as being distinct from what programs already had. Because note, again, it is introducing things that other programs had, right? So things like mating, bottlenecks events, population structure, variation of mutation rate, sex ratios, all of those things existed in other programs previously. These are the 8 things that they thought were new. The first one is that they provide a continuous natural distribution of mutation effects. That's good. You want a continuous distribution of mutation effects. All modern simulators have that. Every single one of them. They gave a user-specified ratio of dominant to recessive mutations. Okay, cool. All modern forward time simulators do that. Mendel uses an infinite sites model. Okay, but can it also use a non-infinite sites model, a finite sites model? Slim does both. You can use an infinite sites model, or you can use a finite sites model in Slim. Incorporates concept of heritability and accounts for environmental variance. You can do that in Slim very, very easily. Mendel uses realistic chromosome structure. You can simulate whole genomes. You can simulate introns, proteins, regulatory networks, sex chromosomes. All of those things can be simulated in Slim. Super easy. It's in the base, literally in the base manual. Mendel allows control of genetic parameters via a graphical user interface. Cool. So does Slim. Mendel provides several forms of graphical output. Cool. So does every other. Do you see what I'm saying? You see what I'm saying? If genetic entropy were a fundamental biological reality, like they suggest that it is, if that were true, if it really was a fundamental biological reality, why would no other program show it? What is so special here about Mendel's accountant that makes it so much more realistic, so much more user-friendly, so much more sophisticated than what I just showed you in Slim? Okay, now we've gone through in exquisite detail this article, the shortcomings of it, and what I think are some historical blunders in it, as well as, dunked on Mendel's accountant, I think, for long enough. And what I want to do now is just sort of review my past critiques of genetic entropy, including the simulations, and encourage Donnie and Chris Rupe to check out my Fatal Flaws of Genetic Entropy video, in which I go through each one of these in a lot of detail. First, which is one we talked about today, there was no original genome. Our genomes are mosaics of ancestors that lived at different times in the past, which means that at every single position, for every single ancestor in your genome, that ancestor lived in a population, right? So there was no first human. That's the point there. So if there's no first human, then there's no first genome. If there's no first genome, then there's nothing to reference your perfect genotype that has since been degrading away from, right? The whole concept doesn't make any sense. My second point from that video is that the empirical distribution of fitness effects include a non-negligible proportion of beneficial mutations. Consistently, they sweep beneficial mutations under the rug as if they don't matter, or as if they're so rare as to be inconsequential. That's just not what we see in empirical systems. And study after study after study, we have found when we do this in the lab, we find far more beneficial mutations than what you might expect. Several studies have found that the distribution of fitness effects is almost centered around zero, with almost as many beneficial as deleterious mutations. And I actually show this study in my Fatal Flaws video, and it's on yeast. Check it out. You can't just ignore beneficial mutations. Third, high genetic diversity is the sign of a healthy population in conservation genetics. If genetic entropy were true, then populations that harbor the most mutations, what Donnie calls multiply mutant, right, harbors the most mutations, they should be spiraling to extinction. Donnie addresses this point, and I want to play you the clip. In my comment, I make this point, and he reads this point, and then he responds to it. I'm gonna keep the comment in the side so that you can see what exactly it is he's responding to, and as you watch it, you'll eventually forget what it is he's responding to, which is why I'm going to have that comment there. So take a watch. This is uncut, unedited, straight from the moment he reads it until he moves on to the next topic. This is how he responds to this idea.
In fact, conservation genetics relies on measures of genetic diversity to determine the health of a population. So the problem is, let's take humans. We say that every single human is multiply mutant. Yes, that's because every single human on this planet has accumulated more mutations than their parents and grandparents. Every human has a complex array of mutations, especially deleterious mutations. And the problem is, and I'll use an illustration, an analogy. Let's say you have a room full of people. Let's say 100 people. Everybody has mutations that they've accumulated. Notice this on the slide. Every chromosome has a complex array of nearly neutral and deleterious mutations. Each chromosome also comprises rare beneficial mutations, but the interactions between these countless mutations means selection is inefficient to solve the degeneration problem. Since we have, as a population, an increasing genetic load, selection simply cannot sort out the good, the bad, and the ugly. And this is the point that we make in the article. Selection is what? Selection is limited. Fixation or genetic drift's not going to work because you're nearly neutral. Your neutral mutation rate over a long time equals your fixation rate. And a lot of your other mutations, they're floating around the population, still degenerating, still leading to disease, and they don't have to drift to fixation to result over time in genetic sickness in whatever the respective organism is. And that's what a lot of these critics, unfortunately, miss out on. And so if you had a room full of 100 people, every single person is multiply mutant. That is a very proper way to describe the human population. 7 to 8 billion people, everybody on the planet has accumulated thousands of mutations. And so let's say we were to invoke really strong, unrealistic selection. Let's remove 50% of the worst mutants, the ones with the most damaging mutations out of the 100 people in that room. Okay, so you remove 50 people. What are you left with? Are you solving the degeneration problem? No, you are not, because you're still left with what? You're still left with 50 people who are still more mutant than the generation before it, because those 50 people still have more mutations, roughly 100, than their parents and their grandparents. And so all you can ever do is slow down the degeneration process. Notice this: realistic parameter settings demonstrates that genetic degeneration is inevitable. This has been published in numerous secular journals. And so those are just a couple points that I wanted to address.
Next is that there is no evidence that populations are uniformly losing fitness, and this includes humans, right? We see plenty of populations that are growing in size, they're constant in size. None of this is what you would expect if there were a sufficient and accumulating load that was uniformly reducing fitness of organisms. Compensatory mutations allow populations to maintain fitness equilibrium. I showed the math for this in the original video, and the simulations show this, which I'm just fixing to talk about. Whenever you apply an empirical distribution of fitness effects, not one that you just made up in your head, not a purely mathematical one, but an actual empirical one based on a study, you don't get genetic entropy. I've sent these to Donnie. I sent Donnie the code for this to show Chris Rupe so that he could run it himself. I literally gave him the code to do this. And this simulation was presented in that video. So let's walk through it one more time, just so we're clear. What I did is I set up a population that's not of constant size, right? It can go extinct. It can rise. It'll fluctuate, right? It's what determines the population size is how efficiently organisms in the population can get resources from the environment. Okay, so there's an environmental component that the organisms are living in. They can't just grow exponentially or they'll exhaust the environment. Okay, that seems pretty realistic. Now, there is random mating, but I want to stress to you, Mendel's accountant assumes random mating. It's not even very clear to me if you can have assortative mating in Mendel's accountant, but regardless, there is no mate choice in any of the simulations they run. Random mate, it's all random mating. So that is a simplistic assumption of this simulation, but it's also one of theirs. I employ an empirical distribution of fitness effects. So what I'm showing here, this little gray bar is a screenshot from the GUI, from Slim's GUI. On the left is an exponential negative distribution of deleterious mutations. You can see most of them, the highest proportion, are around neutrality, right? So they're very mildly deleterious. That's the ones that they're talking about here. And I'm comparing this down here. This is an empirically estimated one from Linsky's long-term evolution experiment. And so you can see at 0, this is where selective neutrality, and then you can see it roughly approximates what I'm showing, right? It's a close approximation to that. On the other end, so going into the positive direction, From theirs, you can see theirs is pretty quickly declining straight down to around 0.1, so around 10% fitness bonus. I pick a very, very conservative beneficial distribution. It's gamma distributed, and it's teeny, teeny, tiny. The vast majority of the selective effects are very, very near neutrality. And you can see this because you can barely see that there's a little tail right here. It's a little blip of higher values, right? The highest density of values that will be picked are very near zero. So beneficial mutations are very rare when they do occur. And when they do occur, they have a very, very slight effect on the fitness. Then what I did is I just plotted the distribution of neutral over deleterious mutations. If they occur, they should be equilibrating, right? You should get roughly the same because the probability of them are roughly the same. And so if selection is inefficient, if selection is incapable of removing them, then that frequency should stay flat. Right, so this is my way of measuring if selection is efficient, and then I plot population size. So right here, this is the population size across 50,000 generations. In humans, that's equivalent to over 1 million years, right? 1.25 million years, I think. A long freaking time. And you can see very clearly the population not only is not collapsing, it actually slightly grew. Kind of equilibrated around 1,200 individuals, which means by 1.5 million or 1.25 million years, individuals were actually a little bit better at extracting resources from their environment, such that they could grow in size, right? Notice just below that, we see this declining rate over the course of the population. This is selection. This is how selection works. It's slowly over the course of the simulation getting rid of those variants, because those variants accumulate early on, right? There's not a lot early on, but they start to accumulate. And then once they reach sort of a threshold in the population, selection sees them and can start weeding them out. And so here we can very clearly see that the proportion declines through time. That's natural selection. That's what it does. So again, they say that selection can't do this. It's inefficient to be able to do this. I show very clearly it can absolutely do this. They say that, my simulations are too simple. It's very unclear to me how they're any simpler than the ones that they did in Mendel's Accountant. Please show me how they are simpler. Give me the specific ways in which mine is simpler than yours. And please don't make it just because it didn't show what you wanted it to show. Because I suspect if it had shown genetic entropy, you wouldn't say, oh, it was too simple. And despite showing the exact same thing, you wouldn't say it was too simple. It's the conclusion. You're just going with the conclusion that you're already settled upon, right? That's not how science works, and you can't hand wave it away. They very often say that we just hand wave things away. I think we have been very thorough here and addressed them at the mathematical level, at both historical and mathematical levels, as to why they are incorrect in the way that they've presented this information and in the conclusion of genetic entropy. Yeah, so with that, we've basically covered the entirety of Rupe and Badinsky. I hope you enjoyed it. I hope you learned something really and truly. I care more about whether you learn some pop gen than I do about a debate with creationists. It really, really matters to me that you came away with some pop gen knowledge. So, next time you see them using things like the mutation load paradox, tell them it's a paradox. If it were real, you'd be dead. You wouldn't be around to make that argument. So it can't be real, right? Tell them if they're going to invoke things like very slightly deleterious mutations, they need to have a mathematical model. And they need to be able to show that humans are inside of that range. And if we are inside of that range, they need to be able to show that the drift load is accumulating and that it won't actually just reach mutation selection balance. That's a mathematical model that they need to have. If you don't have it, why would I take your word? The existing math doesn't show what you claim. So either derive some new math or like throw in the towel. That's the way that it is. And I want to thank both Donnie and Chris. If I've come off snarky, I want to say it's in good faith. We have good fun. Chris, I'm sure, can also, be snarky with the best of them. We know Donnie can be snarky with the best of them. It's all in good fun. They were both kind to me. Chris was kind to me in the comments. Donnie was snarky, but he was kind to me in his response. So, you know, no hard feelings. I'm just giving it the way that it is, my man. So again, thanks everybody for being here. If you have any thoughts, any questions, criticism, comments, whatever, leave them in the comment section. I'd be more than happy to address them. So again, thanks so much for being here, and I'll catch you